Coli. By Austin+Hyle. Escherichia coli. The scientific name for E. coli is Escherichia coli. E. coli are members of a large group of bacterial germs that inhabit the intestinal tract of humans and other warm-blooded animals. Classification domain, Bacteria kingdom, Bacteria. Phylum, Proteobacteria. Class, Gamma Proteobacteria. Order, Enterobacteriales. Family, Enterobacteriaceae. Genus, Escherichia. Species, Escherichia coli, E. coli. Structure and metabolism. E. coli is a facultative anaerobe that colonizes the lower gut of animals but also survives when released into the environment. They are rod-shaped bacteria that possess adhesive fimbriae. E. albertii are non-motile bacteria that produce acid from the fermentation of D-glucose, with gas, L-arabinose, and D-mannitol, do not ferment sucrose and lactose. They are beta-galactosidase positive and fermented D-arabinose, D-fructose, D-galactose, D-mannose, and ribose. However, they were unable to use many uncommon sugars. Habitat. E. coli is the most abundant facultatively anaerobic microorganism that is found in the GI tract of humans and mammals. The average human GI tract contains E. coli in the intestine of a human upwards of 1 kg, 2.21 pounds, of bacteria, and approximately 0.11%, these are E. coli. Special adaptations. 6 lines of Escherichia coli adapted for 2000 generations to a stressful high temperature of 41.5 degrees Celsius were examined on a genome-wide scale for duplication/deletion events by using DNA high-density arrays. Toxins. E. coli isn't usually disease-causing. Sugar toxin is one of the most potent toxins known to man, so much so that the Centers for Disease Control and Prevention lists it as a potential bioterrorist agent. Most kinds of E. coli bacteria do not cause disease in humans. Indeed, some are beneficial, and some cause infections other than gastrointestinal infections, such urinary tract infections, gram-negative. Some strains have genes that cause intestinal infections. These strains, for example, type O157:H7, C gram-negative bacilli, E. coli O157:H7 infection, produce several cytotoxins, neurotoxins, and enterotoxins, including sugar toxin, and cause bloody diarrhea, which, in 2 to 7% of cases, lead to hemolytic uremic syndrome. Function/impact. It is a favorite organism for genetic engineering, as cultures of it can be made to produce unlimited quantities of the product of an introduced gene. Several important drugs, insulin, for example, are now manufactured in E. coli. However, E. coli cannot attach sugars to proteins, so proteins requiring such sugars have to be made in the cells of eukaryotes such as yeast cells and mammalian cells grown in cell culture. Wow! More than 700 serotypes of E. coli have been identified. The different E. coli serotypes are distinguished by their O and H antigens on their bodies and flagella, respectively. The E. coli serotypes that are responsible for the reports of contaminated foods and beverages are those that produce sugar toxin, because the toxin is identical to that produced by another bacteria known as Shigella dysentery. Strains of bacteria similar to E. coli have been found in spent printer cartridges, but only in the cyan ones. Scientists have no explanation